Welcome. From the last video, we are learning about the factors affecting expression of the cloned genes. Today's video, it is regarding the transcriptional termination. The factors which are affecting the expression of the foreign gene into the E. coli, they are the promoter strength that we have discussed in the last video only. The another, it is the transcriptional termination, which is a today's topic. Then there is the plasmid copy number, plasmid stability, host cell physiology, translational initiation sequences, codon choice, and mRNA structure. So going towards the today's topic, which is the transcriptional termination. Now the transcriptional termination in the E. coli or in the bacteria, it has been observed, and it is observed that there are the two types of the transcriptional termination sites over the DNA. Okay, and it is a row independent or the intrinsic termination site. Row independent, it is also called as the factor independent, and the second one, it is a row dependent, that it is a factor dependent. Now going towards the first one, which is a factor independent transcription termination. Now when you look at the DNA sequences at the termination, you find that the recognition, it is due to the similar sequences. The sequences, okay, the sequences which are recognized for the termination of the transcription, they are the similar one, okay, in the number of genes. Okay, and what is that sequences? It is an inverted repeat followed by string of the A residues. And when it is going to be transcribed, then when it is going to be transcribed, then it is how on the RNA, it is on the mRNA, it is a GC rich region. Okay, GC rich region, it is a stem. Okay, it is a stem loop structure, and here it is a string of the uridine residues. It is a single stranded urine. So here in the intrinsic termination or in the factor independent transcription termination or the factor independent transcription termination on the mRNA, you find out that there is a GC rich stem loop structure followed by the uridine residues, 7 uridine residues, and this, it is the signal which is when it has been formed, then there is a termination. So in this type of the termination, there is a no need of any energy, that is, there is a no need of the ATP. Okay, so the energy, it is not used up when there is a termination with the intrinsic termination, that is a factor independent termination. Now going towards the second, it is a factor dependent or it is a row dependent transcription termination. The factor dependent transcription terminators have very little sequence in common with each other. When there is the analysis of termination sequences, it is observed that there is no similarity or there is a very little similarities between the transcription termination sequences. Okay, and second one, it is what termination involves interaction with one of the three known E. coli termination factor, and that termination factors, it is a rho or it is a tau or it is the nus A, or all the three, they are involved into the transcriptional termination. And when you look at these transcriptional termination factors, okay, the factors, this rho factor or the tau or the nus A, these factors, it is utilizing the energy. When you look at the rho factor, rho dependent termination, when you look at the rho, it is nothing but it is a helicases, and as it is a helicases, it is going to use the ATP. Okay, and that energy released by the hydrolysis of the ATP, it is used for the termination of the transcription. So the factor dependent transcription termination, it is using the energy. Okay, it is using the energy for the termination. So when we compare, okay, so when we compare the transcription termination with the factor independent, that is the intrinsic termination, and the factor dependent, that is the row dependent termination, at that time we observe that the intrinsic termination, the sequences, they are the similar sequences in the genes. So we can design the termination sequences easily. When a foreign gene, it has to be expressed into the E. coli. But when you go for, when you go for the factor dependent or the row dependent termination, the row dependent termination, it is utilizing the energy, and the sequences, they are also not too similar for designing the termination for the transcription. Okay, so here what will be the choice? Okay, so what will be the choice when we compare both of them? It is observed that most of the expression vector incorporate the factor independent, that is the intrinsic termination. Okay, it is utilizing the intrinsic termination sequence downstream from the site of the insertion of the cloned genes. Because sequences, they are the similar, so you don't have to much look for which is the better sequence, which is allowing the termination of the transcription. And also, as it is not utilizing any energy, any ATP, okay, there is no ATP hydrolysis, the the gene is accepted by the E. coli, as it is not requiring the energy for the termination of the transcription. So in this way, when there is the choice of the termination site for the foreign gene to be cloned into the E. coli, that site, it is a factor independent termination sequence. Okay, I think you have understood this transcriptional termination. If you like my video, please subscribe my channel and also share my video with your friends, so the knowledge, it is going to spread. If you like my video, please comment, and also if you have any doubt, if you have any comment, write down into the comment box. Thank you.